Okay. So Zach, the floor is yours.
All right, how's my volume in the back? Fine? All right, we're good. All right, guys, my name's Zach Morrisey, like he just said. Today I'm going to be talking more about addiction to you and specifically the things that he just mentioned. But like he said, please hold the questions. I'm just not an expert. I'm not equipped to answer those questions for you. And I know we have a very inquisitive class, which isn't a bad thing, but I'm just not equipped to answer them. So if you have them, hold them. I'll talk about them with you with Dr. Meyer at the end for you. All right. The first thing I'm going to talk about very briefly is just the historical viewpoint of addiction. And like pretty much every single mental health disorder that there is, addiction has been widely misunderstood for pretty much as long as it's been known about. Like depression, like anxiety, we didn't know enough about it to really have a clear understanding. And drug use, the common misunderstanding is that it's just this battle of willpower. Those that are addicted to drugs have something inherently worse about them that makes them more likely to be addicted, and that relapse is seen as sort of this break in willpower, that once you relapse, you no longer have the willpower to stay off of the drugs. So the first experiment that I'm going to talk about with you is this 1970s experiment that was done by a guy named Bruce Alexander. So what he did is he did this experiment to sort of challenge this idea. So what this experiment became colloquially known as is rat park. So what addiction scientists knew in the 60s is that when you took a rat and you put it in isolate housing in what they called a Skinner box, it's just basically nothing other than food, water, and drugs, that a rat will just experiment with the drugs, increase its drug usage, and then eventually overdose and die. And this is pretty much 100% of the time. It'll just experiment with the drugs, use them, and die. And this sort of led to the idea that these drugs are just these profoundly addictive substances that everybody, if they took drugs, would get addicted and end up overdosing and die. But he sought to challenge that, and he thought maybe it's not a function of the addictive quality of the drugs, but maybe it's the situation they're in. Maybe it's the fact that they're housed alone with nobody to talk to. So he devised this experiment called Rat Park, where they had a control group. They had those same rats in the isolate housing, and then they had rats in Rat Park. And what Rat Park was this sort of large enclosure with lots of things for the rats to do, other rats to play with. They actually just cut up a bunch of cans and emptied them out because it turns out rats just like to hide in empty cans, little wheels to run on. It was a pretty good place to be if you were a rat. And in both groups, they gave them access to morphine, which is just an opiate drug. And what did he find? This first graph is the lesser of the two important graphs. On the y-axis, we have drug-seeking behavior, which is just the amount of drugs that they were taking. And on the x-axis here is the sweetness of the morphine solution, because morphine is sort of an aversive taste to rats, and they don't really like it. So as we get further along the x-axis here, this is where they're adding more sweetener to the morphine. And you see here, even when they start adding more sweetener to the morphine, the rats, the black circles, the black squares, that were in isolate housing consumed much more. And the more important of the two graphs is just these choice days, where you can see again that the rats in isolate housing were consuming far more morphine than the rats that were housed in Rat Park were. And if anything, the rats that were housed in Rat Park decreased their drug usage after they first experimented with it. So this was one of the first real experiments that sort of changed how people view addiction. This challenged the notion that drugs are just inherently very addictive, and they will get you addicted, and you will overdose, and you will die. But unfortunately, this study wasn't exactly the gold standard of a scientific study. Because there were all of these rats housed together, male and female rats, they were breeding all the time, so they had a bunch of little rat babies in the rat park, and this made it sort of difficult to replicate. So they had some replication troubles, and he also lost a little bit of his data. So it's not the gold standard of scientific experiments, but it was very important in the ideas that it proposed, and I will come back to a similar experiment later that's a bit more scientific. So this brought two very important main questions for addiction, and these are two that I will be talking about. So the first question is, why do people fundamentally become drug dependent? Why do people compulsively use drugs during drug addiction? Dr. Meyer talked about this a little bit. You have the positive, the negative reinforcement models of addiction. But fundamentally, we're just looking at what are the things that are driving this drug dependence. And what I will be talking about a little bit more than that is why people can't abstain from drugs after they try to quit their addiction. Why do people relapse? All right. So I'm going to talk about the drug dependence first. I'm going to talk a little bit about some neurobiological things that happen during drug dependence. And what we know is that drugs hijack the motivational system of the brain. I know we just talked about that yesterday. That it's not necessarily the reward system, but the dopamine system is the motivational part of the brain. And the brain adapts to drug use. That's another thing that we talked a little bit about yesterday. But importantly, one of the ways that the brain adapts to drug use is that there are decreases in dopamine D2 receptors in the ventral striatum or nucleus accumbens. If I say ventral striatum or nucleus accumbens, they're the exact same thing, just know that. And there's also less baseline dopamine in the brain, which means there's less motivation for the natural rewards. But I'm going to be talking about those D2 receptors. So what we see in addicts and pretty much every single drug of abuse is that over time, drug use is associated with decreases in these dopamine D2 receptors in the ventral striatum and the nucleus accumbens. And what is seen with those is increases in drug-seeking behavior. It's sort of the cyclic thing. The drug use is decreasing the amount of dopamine D2 receptors in the ventral striatum, and this is associated with increases in drug-seeking behavior. And there's the one theory that I'll be talking about is why this happens. So right here on the right, these are from a study done by a woman named Nora Volkov. This is on the bottom we have a meth abuser, on the top we have a cocaine abuser, and they're imaging the activity in the ventral striatum of the dopamine D2 receptors. And as you can see pretty clearly, there is a decrease in both of the brains of the abusers of the dopamine D2 receptors. So why is this causing an increase in drug-seeking behavior? Well, the decreases in dopamine D2 receptors in the ventral striatum are associated with decreased glucose metabolism in the prefrontal cortex. That's just another way of saying decreases in activity of the prefrontal cortex. And when we see those decreases in activity of the prefrontal cortex, we see more compulsive and more impulsive behaviors, more trait impulsivity. So I'll talk about a study that showed that. So the decrease in dopamine D2 receptors, decreased levels of prefrontal cortex activities, increased impulsive behaviors, and compulsive behaviors. And this study shows that. So they measured the trait impulsivity of rats. So how they measure trait impulsivity, they're measuring choice trait impulsivity. There's a couple different types. But choice trait impulsivity, they test that by training rats to know that if they press lever X, they're going to get an immediate but small amount of food. And if they press lever Y, after a short delay, they'll get a larger amount of food. And the rats that are choice impulsive will be less able to press the lever for the delay and get the larger amount of food later. They prefer the instant choice, the instant amount of lower amount of food. And the rats that are choice impulsive, seen by these circles, the darker circles, they escalate their drug taking much quicker than do the rats that are non-choice impulsive. Choice impulsivity, decreases in dopamine D2 receptors and decreases in prefrontal cortex activity are things that are seen inherently among people with ADHD, which is why people with ADHD do show higher comorbidities with drug addiction, especially stimulant drugs of addiction such as nicotine. If you're going to remember anything, remember drug use causes decreases in dopamine D2 receptors, causing decreases in prefrontal cortex activity. And the theory is that this prefrontal cortex activity decrease is resulting in higher impulsive behaviors and higher drug seeking and compulsive drug use. So, if these dopamine D2 receptors are so instrumental in mediating drug use, can we change that? Can we change the amount of dopamine D2 receptors in the brain and also at the same time change drug use? And the answer to that question is yes. This is a study that was done that showed that what we have here is a vector study. What they did is they injected rat brains with the DNA required to make dopamine D2 receptors into the ventral striatum. Remember, DNA makes RNA, makes protein. That is the central dogma of biology. They injected the nucleus accumbens with the DNA required to make the D2 dopamine receptors, which means that area of the brain is going to produce more dopamine D2 receptors. On the x-axis, or on the y-axis, forgot my axes for a second, y-axis of this graph is total infusions. The infusion is just the drug-seeking behavior of the rats, as measured by pressing the lever and getting an intravenous drug infusion. On the x-axis are the days. Here is a null vector. This is just their control. They're injecting some non-active component into the nucleus accumbens of the rats, and you can see it didn't really do anything. But here's the important part. This is the D2 vector. They're injecting the DNA for these D2 receptors into the nucleus accumbens, and they see an immediate and statistically significant decrease in drug-seeking behaviors. All of those little stars on top of the bars there are statistically significant results. So you might be asking, is this an effective treatment for drug addiction? And not really. The first problem is that it doesn't last very long. Most of these studies show that this lasts for only a couple of days. This study showed that it lasted a little bit longer than that, but it's not a long-lasting effect. Also, nobody really wants a giant needle shoved into their nucleus accumbens. That's not, you'd have to go in every week pretty much to have somebody shove a needle into your brain and inject it with DNA to decrease your drug-seeking behavior. It's also very expensive. So while these were promising studies and they showed something important about the dopamine D2 receptor system, it's not exactly an effective method of drug treatment. The things that I just discussed with you are why we can see an increased motivation to take drugs, even if the subjective pleasure of the drugs is decreasing. These increases in compulsive behaviors and impulsive behaviors as a result of this D2 dopamine system are why we can see this motivation increasing, even if a tolerance is developing for the drugs. Here's the things to know slide. From that, what I just said, understand the Rat Park study, how it began to change the understanding of drug addiction, and also how drug addiction affects the dopamine D2 receptors in the nucleus accumbens, what changes that causes in the prefrontal cortex, how those changes affect animal behavior, and how this affects drug-seeking behavior. So now I'm going to talk about the relapse portion of addiction. Talked a little bit about the dependence, talked about that last class, and I'll talk about the relapse portion. I know there was a question last class about why people are relapsing so long after they stopped using drugs, and that is something that I hope to answer for you today. I need to distinguish the differences between these two topics that I'm talking about right here, withdrawal symptoms and incubation of craving. I'm not going to talk much about withdrawal, we already talked about it, but withdrawal resolves quickly, at least relative to incubation of craving. The most strong withdrawal symptoms are seen within the first 7 days usually of stopping a drug. It's drug dependent, but it resolves quickly and they decrease over time. When you are going through withdrawal, you're getting the strongest withdrawal symptoms shortly after you stop using drugs, and then they decrease as you continue through your abstinence period. And they're quite severe. We talked about how alcohol withdrawal can cause seizures in rats. In opiate addiction, a lot of opiate addicts call their withdrawal symptoms being dope sick. A lot of their symptoms resemble the flu, a lot of abdominal pain, vomiting, diarrhea, all sorts of nasty stuff. And treating the withdrawal, as we already talked about, is this negative reinforcement model of addiction. People are continuing to use drugs not because they enjoy them, but because they're getting these very bad withdrawal symptoms. I'm going to be talking more about this incubation of craving, and this is what pertains to that question that we got last class. The incubation of craving are time-dependent increases in drug craving the longer an individual goes into their drug abstinent period. These increases are seen long after withdrawal has subsided, They're seen in humans, they're seen in rats, they're seen in all sorts of animal models. And the thing that I will be talking about is the neurobiological reason this happens, one of which is caused by amygdala activation and its glutamatergic projections to the nucleus accumbens. Dr. Murray talked about the dopamine hypothesis, where after long periods of abstinence, your dopamine increases the largest when you take drugs. That's one of them. I will be talking about that portion. Oh no, that's fine. It's okay. In animals, how do we measure relapse? How do we measure this incubation of craving? So the first thing that they do when they're measuring this sort of thing is they what the heck just happened there? I'm giving away all my information, oh no. Here we are. They train rats to respond to a visual cue or an auditory cue. Maybe it could be a red light, it can be a tone, like a sound that they hear, but they train them to know that when that cue is presented, that there are drugs available via a lever press. All of these rat models are just about pressing levers. That's what it all boils down to. But they train them to know that when this cue is presented, that drugs will be available to them. And then, one day, they take the drugs away, and they present the same cue to them. This is called the relapse test, and they see how much these animals are pressing the lever. In humans, cues work in a similar fashion. A cue for a human could be this neon Budweiser sign that I have listed up there for an alcoholic, a recovering alcoholic. Seeing this neon Budweiser sign is the cue that provokes their drug-seeking, their alcohol-seeking behavior. Incubation of addiction is one of the very insidious parts of addiction because you can make it through your withdrawal. You can make it through the hardest parts of quitting drugs and still relapse. Your cravings will still increase even though you've made it through withdrawal. Even though you are well on your way to recovery, you can still relapse, and it's because your cravings are still increasing. This is a very insidious part of addiction. And this is data from a study that showed that. On the x-axis of all of these graphs are withdrawal days. One is the first day after they quit. They take the rats off of drugs, and it goes up from there. The incubation period is different for all different sorts of drugs. You see, depending on how you measure it, heroin has a shorter incubation period. Cocaine has this super pronounced incubation period where over 60 days, there's constantly increasing cravings for the drugs. And super importantly, even 180 days later, 6 months after these rats are taken off of cocaine, their drug-seeking behavior is still twice as high as it was on their first day. 6 months later, and these are just rats. Heroin has this sort of short incubation of craving period, but that's important. But what's important is that this is 5 hours after they presented the cue. When you shorten the period after the cue, you can see this pretty pronounced incubation of craving curve for heroin as well, which means that heroin, the drug-seeking cravings subside pretty quickly after the cue is presented. But this is seen for pretty much every single drug of abuse, even sucrose. Sucrose isn't even a drug, that's just sugar. Even when you get rats sort of addicted to sugar, you can see an incubation of craving curve for sucrose, where 21 days after they quit sucrose, they're seeking that sucrose after the cue more than on the first day. So why do we see this happening? I'll be talking about the amygdala and the nucleus accumbens. The amygdala is the part of the brain that mediates memories. This is where the cue lies in your brain. When you see that neon Budweiser sign, or you're at the gas station paying for gas and you see that pack of cigarettes on the wall, that's your amygdala activating. You see that, and that memory from your drug usage is activating your amygdala. And then you have the nucleus accumbens. The amygdala connects to the nucleus accumbens via glutamatergic projections, and the nucleus accumbens mediates the motivation part of your brain. So you have the activation of the amygdala, that's the seeing the drugs, and it's connecting to the nucleus accumbens, and that's where you're getting this motivation of drug seeking. Fundamentally, if you increase the activation of one or the other, the amygdala or the nucleus accumbens, you can increase drug seeking behavior. And if you see these increases in activity of these two brain areas over time, you're going to see this incubation of craving curve. And that's what we see. So I'm going to talk about the neurobiology behind those two. I'm going to pick this up because it's bothering me. The first neurobiological change we can see is increased ERK activation in the amygdala. What ERK is an enzyme. It's part of a subsection of the MAP kinase pathway. It's just an enzyme in a protein pathway. And we can see that this enzyme pathway activity is increased the longer a person goes without drugs. The longer that a person or a rat goes without drugs, you can see that ERK activity in the amygdala is increasing. What that means is that if the amygdala is increasing in activity, that we're going to see these cues increasing in reactivity. The cues are going to become more potent. ERK has many bodily functions. It's actually active everywhere in the body. But in the amygdala, it's incredibly important in neuroplasticity and Pavlovian conditioning. And Pavlovian conditioning is basically just a cue-mediated craving for drugs. That Pavlov's dog, where you have the dogs hearing the bell and they're getting the salivating response, it's no different than the recovering alcoholic seeing the neon Budweiser sign. This is mediating that response. The ERK activity is going up the longer that an individual goes without drugs, and they determined a causality for that by pharmacologically blocking the ERK pathway. They blocked this ERK pathway and they saw an immediate decrease in drug-seeking behavior. Talking makes you thirsty. Now, there's another neurobiological change that happens, and to talk about that, I'm going to ask you to recall the latent AMPA insertion from learning. During learning, in the nucleus accumbens, after that long-term potentiation happens, you have the insertion of these AMPA receptors into the postsynaptic glutamate neurons. A similar process happens during extended periods of drug abstinence, but with an important distinction, and I'll make that distinction for you. Again, this is just, now we have an increased amount of AMPA receptors in that postsynaptic glutamate neuron in the nucleus accumbens, but they're different. I'm going to talk about the AMPA receptor for you. The AMPA receptor is a 4-subunit protein. What that means is that there are 4 protein subunits that all come together to make this single AMPA receptor. And there are different types because you can have different combinations of these protein subunits. The protein subunits have these incredibly inventive names. They're just glutamate A1, A2, A3, and A4. Nothing particularly fun about those numbers. But the important thing is during extended periods of drug abstinence, the AMPA receptors that are being inserted into the nucleus accumbens are glutamate A2 subunit lacking. They do not have this glutamate A2 subunit. These are the type of AMPA receptors that are being inserted into the nucleus accumbens during periods of abstinence. The important thing is, without this glutamate A2 subunit, they are calcium permeable. The glutamate A2 subunit is the subunit that causes them to be calcium impermeable. What this calcium permeability means is that they're more excitatory. So as you go into a longer period of abstinence, you're getting more of these more excitatory receptors inserted into the nucleus accumbens, the motivational part of the brain. This is why that's important. The amygdala and the nucleus accumbens are connected by glutamatergic projections. The AMPA receptor is a glutamate receptor. We have glutamatergic receptors between the amygdala and the nucleus accumbens. Here's where that cue is found. That's where the cue is activating your brain, and that's connected to that motivational part of the brain. If you're increasing the receptivity from the amygdala to the nucleus accumbens, you're going to increase the responses and the motivations resulting from a drug cue. Like ERK, they established a causal role for this. They blocked the glutamate A2 lacking AMPA receptors, and they saw an immediate decrease in drug-seeking behavior. And they saw that the decrease was greater on day 30 of an abstinence period than on day 1. We know that this is causing incubation of addiction in the brain. And like we talked about yesterday, there are many models of addiction. You have the positive reinforcement model of addiction, where people are taking drugs to maintain this high that they like. And we talked about the problems that are associated with that. And we talked about the negative reinforcement model of addiction, where people are treating their withdrawal with the drugs. They're simply continuing to take drugs because they're going through withdrawal. And the question that I pose is, can you interrupt that system? Can you change the positive or the negative reinforcement model of addiction by introducing other reinforcers? Because drugs are not the only other reinforcer in the world. Anything you like to do can be a positive reinforcer. Anything that reduces your drug cravings can be a negative reinforcer. Anything that reduces your withdrawal symptoms can be a negative reinforcer. And this is what we think was happening with the rat park experiment. Can other reinforcers decrease your drug dependence? Can these other reinforcers decrease your propensity to relapse? The answer to those questions is yes. So I'm going to talk about rat park, but more scientific. This was an experiment done by a guy named Marco Venero that tested basically the rat park idea in a better way. What they did is they took rats and they trained them to respond to two cues. One cue let them know that they could hit a lever and open a door to a rat friend. Super cool. They knew that when this yellow light, in the case of this study, was flashed, that this lever was active, they could hit the lever, and they could open up a door to play with a rat friend. Alternatively, they were sensitized to QB. They were told, they trained the rats to know that when this red light flashed, that they could hit a lever and get an intravenous injection of, in the primary study case, of methamphetamine. That's why I have our great guy Walter White up here and his 99 or 100% pure blue methamphetamine. To those of you who have watched the show, that means something. To others, just know that they're taking meth. And what they did is they put the rats in their cages and they gave them a mutually exclusive choice between the two. They presented them with both of the cues at the same time, and they allowed the rats to choose which one did they want more. Did they want to play with the friend, or did they want the methamphetamine? This was the fundamental choice that, Marco Venero and this experiment tried to show. And what happened? The rats, even the addicted rats, all of the rats, almost exclusively preferred the social reward. When you gave the rats the choice between methamphetamine and the social reward, they chose the social reward, and they chose it almost every single time. More than that, for the rats that were getting the social reward, when they took it away, they consumed less drugs when the social reward was taken away. And they wanted to see why there were these differences in the rats. So what they did is they still had the control group. This control group was called the forced abstinence group of rats, where they just basically took the drugs away from the rats and didn't provide them with the social reward. In the experimental phase, they had the social reward rats, where the rats weren't taking drugs like the forced abstinence rats, but they were doing so because they were choosing not to. They were choosing to play with the rat friend by hitting that little rat friend lever. And they tested the brains of those two different groups to see if there were differences in them as a result of that. And there were. This picture's a little busy, so the next slide is the graphical representation of what this is. But they used CFOS, which we know is the imaging way that we could see how neurons are active in the brain. The two important parts of this picture are this one and that one. These are taken on day 15 of the abstinence period. Day 15 of the abstinence period, once they're objectively over their withdrawal symptoms. In the social choice experiment, all of those little green tags are pointing to these PKC delta neurons. These PKC delta neurons in the amygdala, that's where this is imaged, are more active in the social choice condition. And in the forced abstinence condition, these little red tags that you see right here are pointing to somatostatin excitatory neurons. And when they tagged them, they saw that they were more active in the forced abstinence condition. This is the graphical representation of that. Remember, this is measured in the amygdala. On day 15, for the PKC delta inhibitory neurons, they saw that they were far more active for the social choice and less of them active, and less of the somatostatin neurons active for social choice. In the forced abstinence, there were more excitatory neurons, there were more of these somatostatin neurons active, and there were less of the PKC delta inhibitory neurons active. Fundamentally, what this means is that the rats that were hitting the lever for their rat friend had less amygdala activation, and the ones that weren't hitting the lever for the rat friend, the ones that were forced into their abstinence, had more amygdala activation. What we've been talking about with the amygdala is activation in the amygdala increases propensity for these drug cues to result in drug-seeking behavior. And that's what they saw. These changes in the brains, the decrease in activation in the amygdala in the social choice rats and the increase in amygdala activation in the forced abstinence rats caused real changes. They did a relapse test. They did the same test that is basically measuring the incubation of addiction. And on the first day of their drug abstinent period, the social choice rats and the forced abstinence rats didn't differ very much in their propensity for drug use. When they presented the cue, both of those groups relapsed. They did drug-seeking behavior about the same. The important difference is on day 15, when they presented that same cue, the social choice rats had a very small increase in drug-seeking behavior, whereas the rats that were in the forced abstinence group had a very large increase in drug-seeking behavior. This is a really large effect size. The fact that it barely increased with the social choice rats and it went way up with the forced abstinence rats, that's a huge effect size. But we didn't even see that kind of decrease in drug-seeking behavior in the rats where they were literally injecting their nucleus accumbens with the DNA for D2 dopamine receptors. This isn't even something that is physically being done. These rats are just hitting a lever, playing with a friend, and they're causing real brain changes that result in real differences in relapse behavior. Another fundamental thing that he found was this wasn't just true for methamphetamine, which was his original drug of choice for the experiment, but it was also true for other drugs, and it was true for heroin. This provides substantial evidence for social reward as a treatment for addiction. This really changes how people view addiction and how scientists have viewed addiction. We are going way ahead, way ahead. Here we are, the things to know slide. So the things to know is what incubation of craving is, how it's measured, and what areas of the brain are involved. Remember, incubation of craving is simply the increase in drug-seeking behavior, the drug cravings, as a period of abstinence goes along, and what brain areas are involved. Know the structure of the AMPA receptor and under what circumstances it would be calcium permeable. Remember that we were talking about this as a function of incubation of craving. The AMPA receptors have specific subunits, and when one of those subunits is gone, they're calcium permeable and they're more excitatory. That's blue for a reason. You should know that one. And also know the results of the social choice experiment. Which groups of rats had what brain changes? What was this, and what was the effect on drug-seeking behavior and incubation? Know those things. It's the things on the slide. So the last thing that I'm going to do today is give you a small history lesson. I know you probably don't want a history lesson because this isn't a history class, but I'm going to do it anyway, and I'm going to get up on my soapbox at the same time. In the 1990s, Portugal had this really bad opiate addiction problem. It was very similar to the one that the United States is seeing today and other countries in the world, very similar to those problems. They had high rates of abuse, high rates of opiate addiction, high rates of overdosing and dying. They had high rates of even diseases that are just associated with drug use, like HIV rates as a result of needle injections, hepatitis rates as a result of needle injections. It was a big public health concern. But They did not originally view it as a public health concern. They viewed addiction as this disorder of willpower. They, instead of getting these people help, they implemented these American-style drug punishments for addicts. They basically started incarcerating more addicts and imposing larger penalties on them. And it didn't work. That's the thing. When they first did these changes in the 1990s, it didn't work. All they did was increase the amount of addicts that they put in prison. And we know that putting addicts in prison doesn't work. If anything, it increases their risk of relapsing and dying. The first 2 weeks out of prison for any addict are the most dangerous weeks for them because they have the highest rates of relapse and the highest rates of overdose deaths as a result of relapse out of any subgroup of people. And they thought, okay, we've done these drug policies, they haven't worked. All we're doing is incarcerating more addicts. How do we stop this? And they did something that was more evidence-based. In 2001, they began these very progressive, progressive and structurally changing drug policies. They decriminalized the use of all drugs. There were no criminal drugs to use. To be clear, they didn't legalize these drugs. It's not like Colorado where you can just go to a weed dispensary. There were no heroin dispensaries in Portugal. They decriminalized the usage of the drugs. They didn't decriminalize the sale or distribution of these drugs. They still locked up drug dealers. But they decriminalized the usage of the drugs. And importantly, they also reallocated all of the funds that they spent on incarcerating addicts on social programs. They started these microloan programs to get addicts into appropriate housing. They started all sorts of support groups. They trained addiction counselors. And importantly, when you were caught with drugs, you were no longer sent in front of a judge, you were sent to a counselor. That is what was a profoundly important part of this. So when they made these changes, when they decriminalized drug use, this thing keeps on going crazy on me. We'll go back. When they did these changes, when they decriminalized drug use, what happened? It really worked. So after 2001, when they decriminalized drug use, they started bettering the lives of addicts rather than locking them up. The drug use rates plummeted. The overdose rates plummeted, and on this slide here, I have a graph of the HIV rates as a result of needle using. That basically saw a tenfold decrease in 10 years. Huge, rapid reductions in drug use and its associated diseases. On the next slide, this is actually a picture of a construction class in Portugal. They had all sorts of different things. They had these construction and pottery classes. I even read about Portugal starting these sort of Pilates classes for addicts. Just social support groups, really. And this was one of them. I thought that was a cool picture. But what did we learn from this? What did we learn from Portugal? What did we learn from these animal studies? The first important thing we learned is that incarcerating addicts is like housing rats alone in a Skinner box. All it does is it increases the propensity for drug use. Locking people up is like putting them in a cage. It's like putting a rat in the cage. They consume more drugs when they get out, and they relapse harder when they get out. And when you start looking at addiction like a mental health disorder, the whole logic of incarcerating addicts sort of breaks down in the first place. It's like incarcerating a person with depression for having a depressive episode. They're not going to be less depressed when they get out of prison. If anything, they're going to be more depressed because they were in prison. It's not going to help the problem. It's a mental health disorder. And the second thing we learn is that social support groups, social reward, functions the same in humans as it does in these animal studies. AA, these Pilates classes for addicts in Portugal, these construction classes, these addiction counselors function in the same way that hitting that lever and getting that rat friend did with the rats. It decreases propensity for drug use. It was a profound change. What we learned is that objectively bettering the lives of addicts, not incarcerating them, not decreasing their quality of life, is what helped them recover from addiction. With that, I talked fast. I went a little bit, I ended a little bit sooner than I had in practice. But with that, I'll leave you with that. I think it's a good way to end the semester because it really brings into the macro why we are here learning about biopsychology. What can this mean for us? And with that, I'll leave you. Good luck on your test on Friday, and thank you.